Many bacteria make a dormant, resistant structure upon experiencing adverse conditions, such as dry conditions or extreme temperature, and this protects them. But different groups of bacteria make different such structures, and so the blue-green algae or cyanobacteria and other groups of bacteria, they have resistant structures which are not endospores. Endospores are resistant structures formed by members of one phylum of bacteria, the Bacillota, although it also can be called the Firmicutes, um, and bacterial genera such as Bacillus and Clostridium, which are extremely important for humans and can cause disease. These can make endospores. Clearly, these endospores are a benefit to the bacteria because it allows it to resist extreme environmental conditions such as high temperatures, drying out, or desiccation, many chemicals which would kill a vegetative cell, large amounts of UV light, extreme freezing, a number of enzymes which could kill vegetative cells, etc. So this is something that allows them to survive adverse conditions. What is an endospore? Well, bacteria can have a protoplasm, kind of the same as a cytoplasm, but we often use that term with cells that have a nucleus. So the protoplasm has DNA, ribosomes, and the other aspects of the cell. The protoplasm is dehydrated, and thus this will protect it from heat. So as something heats, the water molecules will become agitated, but if you're dehydrated, you experience this less. Then around this protoplasm is an inner membrane, which is going to regulate permeability and protect the bacterium from certain chemicals. Around this, there is a modified peptidoglycan cell wall, and when the endospore germinates, this will become the cell wall of the vegetative cell. And then around this, there is an outer protein coat, which provides chemical and enzyme resistance. Within the endospore, a specific chemical, dipicolinic acid, helps to protect the cell and is required for the dormancy period. This can represent up to 10% of the dry weight of the spore. In addition, there are other specialized proteins, including the small acid-soluble proteins, which help to condense around the DNA and help to condense the DNA, protecting it from UV light and specific chemicals. Since spores can survive boiling for hours, autoclaving may be needed for sterilization. Autoclaving involves using steam under pressure, which is hotter than normal steam. And a warning to biology students who might take a work study job helping out in a lab where they may empty out an autoclave. This steam is hotter than regular steam, and this is from someone who once got second-degree burns because he stupidly forgot that when he was helping out in a lab.